Curtin University. Finished up about a year ago out of the ARC Centre for Mine Site Restoration, which is an outgoing ARC centre, a bit louder, that focused all on mine site restoration essentially. And my project within there was the use of the Rutaceae specifically as a difficult to propagate plant family here in Western Australia, and how we can overcome some of those barriers and enable the use of those seeds in horticulture or in mine site restoration as a focus. Prior to that, I did an undergraduate degree at Cowan University. I actually picked up a small grant from the Northern Suburbs branch of the Wildflower Society back in 2016, which funded my master's, which was along a similar line of thought, but using the Ericaceae, woody fruited Ericaceae as another difficult to propagate family. And that really, under Hazel Dempster and David Pike up there, really kind of started me along where I am today. So the stuff the Wildflower Society does and the funding they provide to students, you know, that was a $4,000 grant, and that's taken me all the way up till now. So this was the hat I used to wear, and currently I wear the Curtin University hat and now the PTA hat, Metronet, in a new project that we're commencing there. And ask Danielle or myself all about that. Today I'm going to talk about this topic here, which is overcoming barriers to the use of seed in ecological restoration. It's a pretty hot topic at the moment. It's the seed sector in terms of collection, propagation and supply is really booming. The carbon market, biodiversity market that's coming through, conservation, land management, it's all really centering around plant propagation and seed in a lot of cases. And being so lucky to have all the diversity that we do, there's a lot of problems that really come with it in trying to take what is an agricultural system, putting plants back into the landscape and trying to adapt that to native vegetation, or trying to adapt native vegetation to that. So just a brief kind of overview of what I'm going to be talking about broadly. What we start with typically, unfortunately, is degraded ecosystems that we would like to restore. What we want to get back to is a restored ecosystem. And there's an interaction between kind of restoration activities and what we do to put that community back, and what we call the native seed supply chain, or where we actually get the material required for that. So you start with the restoration plan, set your targets, your prescriptions for planting and soil preparation and all that kind of stuff. You develop a seed sourcing and procurement plan, where we're going to get the seed, what species, how much, how far away, all that kind of stuff. And then you go and collect the seed, or depending where you are and what you have available to you, you could also initiate seed production systems. You can actually farm that seed. This is something that happens in America and Europe and to a degree in Eastern Australia. It's something that hasn't really caught on here. You then need to process that seed, and I'll talk about that a little more as we move on. But you need to get it from your raw field collection into a usable, clean product, for lack of a better term, that can be used in seeding or nursery production or whatever it might be. Seed quality testing, you really need to understand the quality of the material you have. Seed storage, I'm not going to touch on. It's a big topic in itself, and there's a lot that goes into that, but for the sake of this, not today. Seed dormancy alleviation, this is really the ecology. This is my wheelhouse, and my passion around this is what are these plants actually doing in their natural environment. That is what makes them difficult to use. That's always what makes them resilient and interesting and all those kind of things. Seed enhancement, I'll talk more about that later, but it is essentially taking a raw form of the seed and doing something to it to make it more manageable, make it flow through seeders better. You can add herbicides and all kinds of things to these. It's not really what I think the intended use should be, but I'll talk more about seed enhancement. I'm frozen. There we go. And then finally, site preparation and seeding. You monitor, and hopefully eventually you get to a restored ecosystem. That's everything kind of in a nuthouse, but what I want to talk about is the kind of barriers to the implementation of that, and the issues that we're in our research group and in many research groups across the country are trying to overcome. So I'll bundle seed supply and seed processing together. and then I'll jump down and talk about seed ecology and seed delivery. I'm going to punctuate this talk between kind of random photos of plants and seeds because that's fun, and then detail and whatnot. But the one thing I do want to raise is when we talk about seed supply and seed processing, the reason that this is so constrained in many places in Western Australia is because of this here. We've got a lot of clearing. In some cases it's near absolute. In some examples you have a very low access to native bushland and places where you can reliably source seed. We also can't take everything that's available, so you're limited by collection targets and whatnot. So getting our seed collection right and getting our seed processing right and making sure that the material we do take is usable and actually is used successfully is more important now than it ever has been or ever will be. I'm just going to flick through some photos of seeds again before I get back to anything too meaty. This is eucalyptus, Lepoda, really small seeds. All the utes tend to have really small seeds. Most of the Myrtaceae tend to have really small seeds. They germinate pretty easily. They're all typically non-dormant except for a few cases, things like Gotocordia and stuff like that. But the biggest issue we have with a lot of these is actually getting them to something that you can see and use and handle. Melaleuca radula, again, small seeds, that scale bar's 2 millimeters. Acacia acuminata, that's your standard acacia seed. That's what they're more or less all going to look like, various sizes. And they typically always have this aril, which helps with ant dispersion and things like that. Atriplex, saltbush. There's lots of interesting things about saltbush, but I have none. This is one of my favourite plants, Perennial Samosa. I spent the better part of 4 years trying to get this to germinate reliably in my PhD. I couldn't tell you how. And I'll get more into this later, but some flora has really complex mechanisms to ensure that it is going to germinate when it wants to, when conditions are conducive, and getting these systems to play with what we want them to do and when we want them to do them is incredibly challenging. Tylotus, Polystachys, anything that looks like this is a nightmare to work with. Anything that's fluffy, these will have a small seed in the base of them, it's always going to be a pain. It's not going to flow through seeders like you want it to. It's just generally quite often quite low viability. I have an example of this here. 30% of what we have in that collection is actually usable for various reasons. Cymbopogon, again, this hairy stuff, I avoid it where I can. Portotheca, again, anything with kind of spikes and spines and things like that is going to be a pain from the processing, and I'll touch more on some of that later. So what I've essentially just flicked through there very quickly is a rough example of annuals, forbs, grasses, shrubs and trees. And what my colleague Simon Pedrini at Curtin did was he took 131 species that are commercially available. And he basically just looked at the prices of them and tried to put a bit of an analysis behind what exactly are we paying for. And you can see, so on average per species you're going to be paying $1,000 a kilo for seed. But that number is more or less meaningless. A kilo of an acacia is going to be vastly different to a kilo of melaleuca. So when we're buying on the kilo, it doesn't really convey that much sound information. And I'll kind of get to why and show you how. So one of the things that we kind of push is this seed quality testing as a process that has been around forever, that we're trying to formalize a little bit more and put our spin on. And it's essentially just estimating how much seed is there in your seed batch, how much of it is chaff, how much of it is sticks and rocks and bugs or anything else, and how much of it is actually usable seed. Separating out the seed from any other material that might be in there, and we do that on a representative sample of the batch. And then we combine that with what we call a seed quality test or a seed viability test. There's a few ways you can do this. The tried and tested is what we call a cut test, where you just cut it open. Is it firm and white? Does it look good? The other end of the scale is something we call tetrazolium. It's a chemical that stains pink in the presence of a metabolite from respiration. So you can slice the seed open, put it on that, leave it for 24 hours, and if it stains pink, there's a good chance it's alive. There's problems with both of those. What we tend to use is what we call, or what is called, an X-ray analysis. And what we'll do is we'll use that, and I'll get more onto how this works and what it is. But it's pretty good at telling us, how many of these seeds are at least filled, how many of them are empty, and then we can use those other methods to kind of calibrate that and make sure all the seeds that are filled are alive. You take your pure seed unit, you take your viable seed unit, and you just do the percentage on them, and you get to a number. So of your 100% of your seed, what is that actual usable fraction? What is alive, what is in a stick, what is in a rock, bug, whatever. This is just an example of the X-ray. So typically when things are kind of shiny and or, filled and white like this, they're going to be alive, and when they're kind of empty and hollow, they're dead. This is just an example of this process. It's pretty good. It's not too labor intensive, but you do have to count your seeds out and you do have to run them through here. And the time intensity is actually going through and counting them on this X-ray and seeing what's there. So one of the things we're trying to develop, and we're trying to speed this up, is basically image recognition and training algorithms on images that we calibrate, which can identify the outline of the seed and then use pixel intensity to tell you what's there and what's not. And this will automatically, on the bottom, spit you out that says 115 seeds recognized, 60, whatever, this many alive, this many dead. And that's far better. You can see that a lot of the ones that it's picked up, I can't tell the difference between. So it's actually using a quantifiable pixel intensity from the X-ray to make that judgment itself, and we calibrate that and check it over time and train it against a large number of images. And this is actually being further developed now to use pixel intensity to give us weights. A lot of the time we spend weighing out seeds and representative batches of big seed batches, so it's a time saver in general. When we do that to those seed batches that we were purchasing before and doing our stats on, what we find is generally across 131 species, about 50% of what we're purchasing is usable for the purposes of restoration. And when we start talking about direct seeding with agricultural machinery or hand broadcasting or whatever it is, that number becomes really, really important. And I'll touch on that towards the end of the talk. Sometimes what's in the seed isn't actually seeds. I always show these because I really like it. This is one of the things I picked up in my master's. This is Conostephium pendulum, Conostephium minus. It's a member of the Ericaceae, really beautiful little tubular flowers. And what we find is actually a native wasp, a chalcidoid wasp in the genus Megastignus, I think, as far as I got through the ID. No discernible entry wounds, so these things interact, I suspect, with the pollination biology or very early on in the fruit development when it's still soft and green and they don't leave much of a trace. And they go through their full developmental cycle inside these things. I kept these in the lab for 3 years and never saw one of these things actually emerge alive. So something, periodic rainfall, wetting and drying, breaks down this hard woody fruit in the field and actually allows these things to come out. They predate about 50% of seeds within these fruits, so they obviously have a big effect on that species, but it is very dominant in much of the landscape, so it's obviously something that's evolved over a long time to work, I guess, like a better word. That kind of takes me into the ecology of it a little more, and like I said, this is really my passion, is kind of understanding what these things have evolved to do, why they've evolved to do them, and how we can actually use them rather than fight against them. You'll hear me talk a lot now about seed dormancy, and you might have quite often heard it spoken of as something that needs to be overcome or broken. We break seed dormancy, and that's a valid approach for use in restoration. You want a seed and you want to know exactly when that seed's going to go in and when it's going to come up. But these are mechanisms that can be used a little bit better to more inform when we seed and how we seed and what exactly we need to do. The way it basically works in Southwest Australia, all this complex dormancy that I've been battling with for all these years, it all basically comes down to this graph, really. Here in the Southwest and in Western Australia, in January, soil temperature is going to be high, it's summer, and over the year it's going to do this. It's going to drop and then it's going to go back up. And soil moisture is going to follow an opposite trend. It's going to do the opposite thing. Most of our seed is dispersed in this kind of late spring to summer period. And if you're a seed entering the soil at that time of year, if you respond to a summer rainfall event, a thunderstorm or something like that, and germinate, you're going to die. There's not enough water to basically keep you going. So all these mechanisms, all this seed dormancy has really evolved to ensure that seeds are going to wait until periods conducive for germination and establishment, which for us is kind of May, June, July, when your rainfall starts to pick up. There's really 2 or 3 things that happen between seeds leaving the plant and seeds germinating. You can sit around in soil dry and typically quite warm, and that's something termed after ripening. Like you ripen fruit on the counter, I guess, is where that term probably comes from. Warm stratification just means warm and wet. And cold stratification means cold and wet. And quite often the thing that we find that works, which I'll talk about, is not this or this, but the change between these. All of these things can, and in many cases do, interact with what we call germination stimulants. And this is things like your smoke, heat from a fire, soil disturbance, light, all those kind of things are something that we say stimulates germination. For many species, if you take a seed fresh off the plant in summer and you treat it with smoke water, there's a high chance it won't germinate. If you give it what it needs and you break that dormancy and then treat it with smoke water, it very well might. And it's just that mechanism and the difference between dormancy and germination that could be better understood in broad-scale propagation and restoration. When I talk about seed dormancy, there's 5 types and 24 subtypes, and it's a hell of a thing. But there's really only 3 that we're really interested in. Physical dormancy, this is the one you overcome with hot water typically. This is something that you're going to see in a lot of acacias and things like that. And then there's morphological dormancy, which is essentially just an immature embryo within the seed that just needs time at adequate temperature and moisture to grow before germination can go forth. I'm going to talk about this one because it's kind of more prevalent. I'm not going to talk about this one today. But physical dormancy is something that we've come up with kind of backdoor methods to break. Boiling water for 3 minutes, that's not something that's really going to occur in nature. People often attribute it to passage through the gut of an animal. That's not really quite there as well. So the example I'm going to use is Cochleospermum fraseri, or kapok. The fluffy material produced in these seed pods was actually sold as bedding and pillow stuffing for quite a long time in Australia, before they found out it causes asthma or something, and that stopped, but that was back in the day. This is a really widespread species across the top end. It's required for restoration of the Ranger uranium mine, of Argyle diamond mine, and we've been engaging with some of the seed collectors and some of the Aboriginal organisations dealing with this species, because they were having a hard time getting it to come up in restoration. It's massive, widespread through the landscape, but they're having a really hard time. It's got this near bulletproof seed coat that restricts entry to water. And that's all physical dormancy is. It's imposed by a water impermeable layer in the seed coat that restricts the passage of water. Many species, all your acacias, most species with physical dormancy have what we call a water gap, or an actual physical site that water will uptake when conditions are conducive to permeating that plug, which is what hot water does, but in many species it doesn't. And for this species, hot water doesn't touch the sides. It'll kill the seed before it reaches that plug. What we did find that works was about 3 hours in concentrated sulfuric acid. Kind of targeted this plug in just the right way to kind of let it go. They could take that for about 7 hours before that started to kill them. And we use a dye to just confirm that that is actually the site of water uptake, and it's not the best image, but it's coming in here and it's not coming in through that seed wall. Once we break the dormancy in these things, once we get past that block to germination, we can begin to look at, well, how do these species recruit naturally in the landscape? How do they respond to temperature and water? Which looks something like this. So this is basically just a scale of water stress. We use chemicals to induce a greater degree of osmotic stress in Petri dishes. So this is an example of no water stress, low, medium, and high water stress. And this is just the temperature profile. So lots of Petri dishes at lots of different temperatures at lots of different water stresses. which gives us this profile of, okay, how is the species going to respond to dryness? How many days with this much water in the soil does this thing need before this proportion germinates? This is where we can begin to get really sophisticated about how we understand species, how we understand their hydrothermal thresholds, and how we use that to determine where are the best sites to seed certain species, what are the best time of year to seed certain species, where do we need to look for new populations of range-restricted species, things like that. So we can actually take our temperature and water potential gradients, we can model them out, and then we can model them across the landscape, and we can look for areas that are kind of more conducive to this species being there or not. This isn't a bulletproof kind of method, but it's something that is being developed, and it's something that is going to be informing broad-scale restoration in years to come. This is all good when we can kind of get things to germinate, and we can make things work and do what we want, but some plants have really complicated dormancy mechanisms that are really cued into multiple cues or multiple things, and that's really difficult to understand in a lab context, and it's even more difficult to kind of put into manufacturing processes and put into things that can actually be picked up by industry and used. So the group I'm going to talk about briefly here is the Rutaceae. This is the group I did my master's on, my PhD on. They take up water readily. They don't have that physical dormancy that I was talking about. They have these big, well-developed embryos. They don't have that morphological thing. What these have is what we call physiological dormancy. And this is essentially imposed by hormones that are produced on the mother plant and then fed to the seed, and they establish what we call a dormant state. It's a big, complicated thing that I'm not going to go into, but essentially what happens is this hormone promotes dormancy, this hormone promotes germination. They respond to environmental signals. So warm, dry is going to promote this and deter this. Cool, wet is going to promote this and deter that in a rough sense. And when that hormone concentration shifts, seeds are non-dormant, and smoke and whatnot can stimulate their germination. And as our season cycles back into those warm, dry, this increases and seeds go back into dormancy. And they do this year in and year out for many species. Don't need to worry about that. What we do need to worry about is that this is a very prevalent thing in our landscape. I ran an analysis, which is in prep, on basically how many species with physiological dormancy can we not propagate for the purposes of restoration or horticulture. I produced a big list, used my supervisors to check it, and then basically went and hassled various nurseries to say, can you grow these species? Is this difficult? Is this not? Kind of fact check myself and put this thing together. And it's basically how many species within an IBRA region and IBRA region are difficult. Or are not difficult, but we don't understand enough for the purposes of utilizing them and breaking that physiological dormancy. It is pretty much one-to-one correlated with diversity. I'm not going to, we could talk about that for days and months. But depending where you are, there's going to be a higher proportion of species that we call intractable or difficult to work with. The old term for that was recalcitrant, but it's changed. But at the end of the day, all it really comes down to is, these environmental processes, which are quite simple on paper. But when you start to think about day-night cycles and hot, dry, and wet, it's infinitely complicated. There's too many combinations to really test effectively, and I've tried a bunch of ways to, test different combinations of soil conditions and temperature in the lab, and full big factorial experiments and soil probes and a million petri dishes to try and figure out what the hell's going on. And in one case it works, and when it does work, it's quite a simple thing. So in the Rutaceae, and the example here is a Phalabium and a Diplolina, about 4 weeks at 30 degrees wet before moving them to cool temperatures really is what they were looking for. It's as simple as that. Not massive germination results, but more than anything that was previously possible. So what this is showing essentially is 0 weeks, 1, 2, all the way up to 12 weeks at 30 degrees wet before moving them to 5, 10, 15, 25, 30, 35 degrees Celsius and trying to see where their optimum kind of germination temperature is. So all we're really interested in here is this peak of the curve. And as these things spend more time at warm conditions, the peak of this curve actually shifts to the left. So fresh seeds straight off the plant germinate best at 25 degrees Celsius. If they go through this warm period in the soil, which is present kind of towards the break of the season, you get a bit more rainfall, things are starting to, it's still summer, but the soils are getting a bit wetter, you're going to get this warm, wet storage coming. And the longer that period goes on for, the cooler the temperature that these things are going to seek for. And this is what we call risk avoidance. These things are experiencing the shoulder of the season, and they're lowering the temperature to seek further into the season, further into this cooler, more conducive period for germination. Or they can do the opposite. So depending what these optimum temperatures are doing, if they're going up or they're going down, we can put them into boxes and classify them as type 1 this, type 2 that. It's more or less irrelevant for the purpose of this. But what it does let us do is box species up and put them in the context of the environment. So a type 1 species that is increasing the germination temperature on the break of the season is looking to germinate early in that season. It's increasing its optimum temperature. It's trying to take advantage of early season germination and gain competitive advantages over other things. Or is it lowering that temperature and seeking to bet hedge essentially and avoid drought mortality in the early season and seek better conditions further on? That's more or less what I do day in and day out. It's just lots of petri dishes and figuring with temperatures and then trying to figure out what the hell's going on in as many species I can get. And if we can build these profiles for entire communities of species, we can start to understand, well, what are the thermal tolerances to climate change, to drought? How far can we push these things before recruitment is unlikely in our restoration sites, in our conservation reserves, whatever it might be. Moving on to the final little bit is seed delivery. So once we've broken the dormancy, or once we've not broken the dormancy, because sometimes it's better to not, sometimes maybe you can just put these things out there and let nature take its place. But once we've come past that point and we're ready to go, how do we actually deliver seed into the environment? So we can do it manually, small areas, limited funding, things like that. You can just take seeds and throw them out. Or you can get mechanical about it. You can use a tractor or a drone or a bull, whatever you have available to you. And these have different uses and different success rates depending on what you're doing. So seed broadcasting, which is typically just chucking seeds out into the environment. You don't know really how many you're putting in location A or location B, or how many are getting to the depth in the soil they need to get to. Generally it means they're going to be more susceptible to desiccation and predation and blowing away with the wind. And at the end of the day you're going to end up with a waste of seed. Or you can take the other approach, which is precision seeding. You can use drills, canola seeders, things like that, and actually target seeds at specific depths that are going to be more conducive to their own ecology. As a general rule, the bigger the seed, the deeper it can emerge from. More resource for it to draw upon to grow. So we definitely recommend, where you can, precision seeding using equipment. What it lets you do is have a much greater control over your seeding rate. And this is where I come back to our pure live seed and actually understanding exactly what is present in your collections, so you can do things like this. So let's say our restoration target is 5 plants per square metre. You would assume we need 5 seeds per square metre. There's a whole way of actually converting seeds to plants. Yeah, a few things there. So let's assume we want 5 seeds per square metre. Well, hypothetically, how many seeds are going to reach the appropriate depth? It might be 20%. Say it's 1 seed. How many of those seeds are going to germinate? How many of them did we not break dormancy in? How many are at the right temperature, or was it raining at the right time? There's always going to be a failure rate in that. So we can say 80% of the seeds we put out there germinate. How many seedlings emerge? The biggest constraint to successful seeding is actually emergence. So that time when a seed is committed to germination but hasn't really established as a small seedling yet is possibly one of the more vulnerable times in their life, and it is where we typically get the most attrition. So say 40% get there. How many plants survive? So of those ones that do emerge, well, how many are actually going to turn into plants? Say it's 50%, hypothetically. So of those 5 seeds or 5 plants per square metre and 5 seeds that we wanted, we actually need about 156 per square metre to get to where we are, which is it's about a factor of 30. That's where really understanding what we're using and how we need to adjust our seeding rates. It's not to say that we need perfect seed collections, but it's to say if we know that 50% of this is unusable, you can leave it in there, you just double your seeding rate. And then you, again, double and account for all these once you understand them in the system, or do things to reduce failure throughout this system, which is where we come to these. Seed coding is not my real wheelhouse, but it is a good silver bullet, or it's looked at as a silver bullet in the industry quite often, like drones were and like, you know, many things are. All it is putting things around the seeds. People tend to gravitate towards, well, can we put growth hormones in there? Can we put herbicides in there? Can we put fertilizers in there? And we definitely can. But by and large, most of those things aren't going to replace broad-scale agricultural practice in the right place. What these things really do help with, what this pelleting really helps with, is standardizing seed size, standardizing seed weight, standardizing density. So we can take something as small as a melaleuca and bring it to the same size as an acacia or whatever else we have. We can standardize a seed batch, and instead of manufacturing specialty seeding equipment, we can just use a canola seeder, and we can use agricultural equipment that's already available and one-pass diverse species mixes across the landscape. The process uses essentially a cement mixer and a bunch of inert glues and clays and powders to just build up these little coats on the seeds. Like that. You end up with a big increase in volume, but by and large it has no effect on germination as we do it. Slightly better or slightly worse depending on the species, but nothing significant. Yeah. More or less I've kind of run around this circle here. There's a billion other things that could be covered in a lot more detail or a lot less detail depending on what you're more interested in. Please come and talk to me if you have anything specific you'd like to flesh me out on. I can see I've accidentally left a random slide of photos in here. That's fine. These are seed embryos. That's all.